So when we actually established the model, this was quite new model. There was no iPS-derived alveolus-on-chip before, and we were quite curious to see if actually influenza virus will replicate in our model. And we also we put an accent on our immunocompetent model. Hi, my name is Hristina Koceva, and I'm a researcher at University Hospital Jena within the group of Alexander Mosig. My research focuses on establishing a human iPS-based alveolus-on-chip model for studying respiratory infections. My motivation behind this research project was the lack of the human-relevant lung in vitro models. As we know, the 2D models are not complex enough to depict the whole complex alveolar environment, and also when it comes to animals, many experiments has been done and many things has been discovered. However, anatomy and physiology of mouse, for example, is quite different than the one in humans. So translating everything can be a bit challenging. So we designed our chip on a way that we use the membrane as a scaffold for seeding of the cells. So we have the epithelial cells on the upper cavity and endothelial cells on the lower cavity. And we have liquid phase and air phase, which is quite important actually when it comes to lung research. So we have the peristaltic pump and the liquid phase on the bottom, and the cells are exposed to air on the upper cavity. The way how we actually co-culture the model is first that we differentiated. So we have a fully isogenic model. So we have one cell line, iPS cell line, and then we differentiated into endothelial cells, monocytes, and epithelial cells. And this is our cell source for co-culturing of the chip. So here briefly we have some more details about how we actually establish the chip. So first we have the seeding of the upper, then of the lower cavity. Then we added the third cell type, the macrophages. We have the peristaltic perfusion, exposure to air, and then our model is ready for treatments or infection. So when we actually established the model, this was quite new model. There was no iPS-derived alveolus-on-chip before, and we were quite curious to see if actually influenza virus will replicate in our model. And we also we put an accent on our immunocompetent model. After optimization and characterization of the infection in our organ on chip, we wanted to see the effect of the macrophages. And when we stained the tissue that was with absence of macrophages, we saw that the viral replication was higher compared to the infected sample. Next step was to check the effect of butyrate in our pre-treated lung tissue after influenza infection. We made our readouts are pretty much immunofluorescence and plaque-forming units, which showed that after butyrate treatment, we had less viral replication in the system. The most interesting findings were that during the influenza infection, our model expressed a higher level of antiviral interferon-related pathways, but on the other hand, we had these pre-treated samples that actually showed that there is a decreased metabolic activity of the cell, which is not the best environment for the influenza infection to replicate. So this can justify our decrease of viral titers and also decrease of nucleoprotein stainings in our previous results. So first of all, trust the process and have a clear biological questions that you want to answer. Don't just choose the tool, but make the whole idea of using organ on a chip to have a specific answer for your project. It can have, science can be quite crucial, can have really ups and downs, but believe in yourself, trust the process, and the results will come out.